I've lived with me the whole life, so I'm always surprised when other people don't know who I am. Okay, I'm speaking about our foundation today. This is the Paul H. DeRay Pathology Fellowship Fund. If our mission statement is here, we have vowed to do just pathology, human pathology research on tick-borne illnesses and look for infectious etiologies of Alzheimer's and other neurological diseases like multiple sclerosis. My personal interest in starting this was because of multiple sclerosis. And we also have in our statement to do cause and effect research. We want our research to have direct applications to patients. We don't want to get bogged down in minutia. We're not going to be the ones that look at the amino acid sequence of a certain protein and compare it to another protein and just completely lost in that kind of thing. We want our research to benefit patients eventually, if not sooner. So, you know, we still have 5 big problems out there that we're still being told by the medical experts and are still being reported in the newspaper and such. And I've run into these almost on a weekly basis locally, but we still hear that it's not transplacentally transferred. As late as 2006, Gary Wormser of the IDSA said this in a film. He's never seen any evidence. And all of these things, I'm going to show you evidence tonight that really disprove all of these myths about Lyme disease that are still perpetuated. The serology tests are 90% accurate, and especially in chronic late-stage neurological disease. If you go on the CDC website, it says that in neurological disease, it's 100% accurate. I just don't see how they can say that with what the data is showing us. That it's not an intracellular disease. I'm going to show you so many examples tonight that this is primarily an intracellular disease. It's what's causing all the problems with persistence and the inability to completely eradicate this organism with antibiotics. It does not persist post-antibiotics. Tonight I'm going to show you a case from Twig, Minnesota of a logger who was sick for 30 years and was treated for almost 10 years. He was treated continuously for 7 years with very aggressive combinations of antibiotic therapy, and we still found 3 species of Borrelia bacteria in his body, 2 in the brain, and then we'll talk about the final case at the end of this lecture. And does not cause dementia. Well, I mentioned to an earlier group here today that in 1903, there was a big argument in the medical literature as to whether syphilis, a similar spirochete, caused dementia in humans. And it went on for 10 years back and forth in the medical literature. Finally, in 1913, it was decided through pathology, brain autopsies, that syphilis did in fact cause Alzheimer's, or a dementia, which they called general paresis. Now, 100 years later, we're in the same exact argument as to whether Borrelia causes dementia, or more specifically, Alzheimer's dementia. And this argument has been going on, and despite the pathology evidence that we show, you have a speaker coming up in October, Dr. Judith McCloskey, and she'll probably address this, but her work goes back almost 15, 20 years, and to deny that Lyme disease gets into the brain and to deny that it's causing dementia-like states, I think is just either complete denial or naivety. So we're going to address some of those topics tonight. We're going to look specifically at the work of Dr. Alan McDonald, mostly from 2013 through present day. Dr. Alan McDonald is a retired pathologist. He lives in Florida and has taken this seriously his entire life. He has some of the best early research going back to the 1987, and still, you know, in 2016, nobody can touch what he has done under a fluorescent microscope. And I think you'll be excited to see some of those findings. Clarity in illumination. We want to add clarity to this very convoluted and difficult to understand disease. But to do that, we needed better tools. These are all working, right? Okay. And we needed better resolution. And I'm going to show you some examples of how these tools do give us better resolution. So we developed on our own something called DNA beacon probes, also known as fish stains. The acronym for fish stains is fluorescent in situ DNA hybridization. We take specific segments of DNA and we put it into a loop, and at the end of that loop we have fluorescent dye. And when this DNA sequence that's specific for the species of Borrelia that we are looking for comes in contact with that species in the human body, in this case brain autopsy material, those fluorescent dyes open up and they will only fluoresce when you hit your target. And this has been shown over and over again. We're not the only ones using fluorescent beacons, but we're the only ones doing it in Lyme disease in actual brain tissue. We're looking at actual human studies. And we're going to show you, I've been registering patients for brain autopsy since 1997. And of course, people don't die on cue, thankfully. And I got to know a lot of these patients over the years. And the one death that I'll focus on, I knew Scotty for forever, and I knew his brother for even longer. And what a tragic tale that is for the family. The fish stains are very useful to us for looking at spirochetes in particular. So you can see this is the DNA molecule. If the entire genomic sequence of the Borrelia spirochete, that's what makes up the spirochete, there's over 300 copies of that that's embedded in two compartments, the outer membrane of the bacteria and also the inner peptidoglycan layer, which we call the cell wall. We use stains that can detect DNA in both compartments, and it gives us almost a three-dimensional picture of the bacteria. But because it picks up this DNA that's scattered throughout the entire Borrelia structure, we get great outlines of spirals. And other fish stains, if you're looking like the DNA for E. coli or something, you just get little microscopic flecks. We get the whole picture. So back in the day, similar detection was PCR tests, but PCR tests did not give you the picture. So this is like the days of radio are past, and now we're looking at the days of TV. We get the picture and the species all in one slide, but we're using this specifically on tissue samples. But I'll show you a couple instances where we used it on blood. Here we're trying to show the compartments of the Borrelia with an electron microscope. That's not our work. So here's our work, though. This is from the hippocampus region of an Alzheimer's patient who died. I think this patient, does it say his age, 80? And you can see that we've stained for the cytoplasmic DNA and also the outer membrane DNA, and it gives us almost this 3D picture of the bacteria. Now, we get a lot of comments from people like from Yale and just recently, as like 3 days ago, Mayo Clinic, saying, well, we don't know what you're looking at. Mayo Clinic claimed that what we're looking at are the edges of human cells, not the Borrelia. So here, we took, going back 3 years, we've done control studies. This is Borrelia burgdorferi, the Lyme bacteria that we grew in culture. Then we took our fish stain that was specific for Borrelia burgdorferi, and we stained it. And sure enough, we get this nice shape of a spirochete from the culture. Now here, you know, in culture, of course, our stain was absorbed. It's very dense, so we get this ultra illumination. But here, we took a culture of Borrelia burgdorferi, and here we have like 5 or 6 spirochetes lumped together, and we used our Borrelia fish stain, and we stained it. But down below, this is Borrelia burgdorferi found in the blood of a 16-year-old boy from Connecticut. And that's a tragic story. At age 14, he got Lyme disease. He tested positive. He was treated for a few weeks, like 4 weeks. Then he relapsed, and 2 years later had a dementia-like state. They said it couldn't be Lyme disease because he had been treated, and by definition, he was cured. This is Connecticut. This is Connecticut. And at age 16, he succumbed, and we did the autopsy on the brain and blood. And we found not only Borrelia burgdorferi, but we found another Borrelia that we're going to talk about that has an attraction for the human brain. And it's Borrelia miyamotoi, a relapsing fever bacteria, and it's found in the same exact tick that transmits Lyme disease. Now, this is Borrelia burgdorferi. In our early tests, this is grown in culture. And once again, we wanted to try our fish technique to pick it up. And as you can see, we're getting individual spirochetes, and we tried a blue dye. We have tried blue, yellow, red, green, and violet. And we have found by far that the green and red stand out the best. But we have relegated the red stain specifically for Borrelia miyamotoi, the green stain for Borrelia burgdorferi, and blue stain for Borrelia mayonii. We're, as far as I know, after speaking with Bobby Pritt from Mayo Clinic, we're the only ones that have a fish stain for Borrelia mayonii, and she didn't seem too happy about that. So I wrote a paper and posted it on our website, the Paul H. Duray website, called The 7 Provocative Findings of Alan McDonald since, well, we're going to go back to 1987, but I think that most of the work that you'll be interested in was done in the last 2 years. So the first, Borrelia burgdorferi crosses the placental barrier. And this is an old slide from 1989 done at Stony Brook showing the Borrelia cultured on umbilical vein, human umbilical vein. And you can see that there's a hole here where the bacteria can now leave the bloodstream and enter a tissue. On the other side here is where that bacteria is going to end up. Is it going to end up in the brain, the heart, the liver, the stomach? Well, it's kind of random, but the bacteria seems to want to leave the bloodstream. Unlike other Borrelia, Lyme disease does not like to stay around in the bloodstream, at least in this form. And we know that it can enter the blood vessel cells, called endothelial cells, and they can sit there and they can live there intracellularly. So the people that say this is not an intracellular disease, I won't name any names, but I want to. Okay, those that say it's not an intracellular disease, the very first human cell it comes in contact, it enters and lives there and can stay there and can move on. And that was shown all the way back by researchers Ying Ma back in 1989. So how can you say there's no evidence? I mean, the evidence is all over the place, especially with endothelial cells. Alan McDonald in 1989 did 7 consecutive fetal autopsies, and he showed very clearly the bacteria can be found in the umbilical fluid. Well, that's a bad sign. You don't want it there. Here it's in the placenta. Well, we're getting closer to the fetus. Here it's in the fetal brain, and here's a slide showing the fetal brain and the placenta. Here it's in the fetal kidney. In adults, we don't often find it in the kidney. And then, lo and behold, here's the spirochete half in and half out of a neuron in the brain of an infant. So that tells us that this is not only intracellular, but it gets into the brain and it can penetrate these cells. And I'm going to show you a very good autopsy from 1997 in adults that shows this better. In this particular case in 1987, a California woman had a diagnosis of Lyme. She had many late stages for Lyme, but she was seronegative based on the Yale Medical Center serology testing. She was not treated with antibiotics. She gave birth to a stillborn. Live Borrelia burgdorferi was cultured from the fetal brain. And this is work done by Paul Lavoie and our namesake of our organization, Dr. Paul H. Duray. A pathologist that did a lot of Lyme disease research in the early 90s. Now, To deny treatment was one thing, but the fact that it was cultured, you know, there's 3 layers of proof. You've got blood tests, but a step above that is pathology, which I'm going to show you, and then a step above that is culturing and identifying the bacteria from culture. And when you get to that stage, if you don't accept that, then you're really in a state of denial, and you have to have a reason or an agenda to deny that kind of evidence. And here we had it in 1987, and Yale was part of that. 1989, 24-year-old woman, positive serology, but negative by Yale ELISA test, gave birth to a stillborn. The bacteria was cultured from the following organs: the fetal liver, the adrenal gland, the brain, and the placenta. But this guy, and once again I'm not going to name names, said he has seen no evidence of transplacental transfer of Lyme disease as late as 2006. So, you know, what do you want to call that? I call it denial. I think that our evidence done by the man I work with, Dr. Alan McDonald, has been conclusive over and over. I mean, I showed you 7 slides. He has 700 slides. Okay, the second provocative finding by Dr. Alan McDonald is that Alzheimer's patients often have Borrelia bacteria in their brain, but it's not in the way you think it is. You would think it's there, it's entering the brain cells, it's irritating the brain cells, but the discovery that we had is pretty eye-opening. Now we're going to go back to the first brain autopsy that I was involved with Dr. Alan McDonald, and this is a patient I registered from Ashland, Wisconsin, and a lot of you have already seen these slides either here or on my website. But we see intracellular Borrelia burgdorferi inside brain neurons of this Alzheimer's patient. And here we see more pictures. Here's a close-up of a neuron with a spirochete half in and coming out the other side. There were also extracellular spirochetes between the brain cells. So this is indicating that this entire cerebral cortex had Borrelia burgdorferi, complicating this case of Alzheimer's. At this point, of course, we're not saying it caused the Alzheimer's, but it's there, and it's something that, you know, it's not doing anything, it's not benign. Now, in 2006, the Dr. Jill Livengood and Robert Gilmore of the CDC did their pathology test, but it wasn't on human cases. It was, they grew human brain cells in the petri dish. And here they grew a neuron, and the green spirochete here is Borrelia burgdorferi that they took from culture, and this is a B31 lab strain. And within hours, it attached to the neurons, penetrated the neurons, and this yellow one is inside the neuron. You're going to get a better picture here. This is a cerebral cortex neuron with intracellular Borrelia burgdorferi. And their paper shows about 24 instances of Borrelia penetrating neurons, another cell in the brain called the glial cell, which helps repair the brain, and also endothelial cells, which are the cells that line all of our blood vessels. So if it can penetrate blood vessels, it can get to the brain. If it gets to the brain, it's like a smorgasbord. It chooses the glial cell or the neuron. But now their study that you're looking at here, that's done on a cultured cell. This next slide, from 2013, I believe, is what Alan McDonald found in a human neuron in an Alzheimer's patient. And this is intracellular Borrelia burgdorferi inside a human neuron from an Alzheimer's patient. So we went from the experimental of the CDC to the actual proof in the real world. Now, at this point, we get people saying, well, how do you know what we're looking at? Do you run controls? And we certainly do. This is Borrelia burgdorferi cultured from Alzheimer's brain tissue and identified as Borrelia burgdorferi. And some of this work was shipped out, so we didn't do our own controls. We had it corroborated by other labs. Now, what did I say was the highest level of confirmation of proof? It's culturing. and we cultured this directly out of an Alzheimer's brain person. Now, we were not the first to do that. Judith McCloskey did that way back, gosh, I want to say 1999. I don't know the exact date, but it's already been done. We've already proven this. Why do we have to keep proving it? But apparently we do. This is from 1929. This is a general paresis patient who had syphilis that caused dementia, and we see these colonies of syphilis bacteria. And the question is, do we see anything like that with Borrelia? Then Judith McCloskey comes along and sees these things, and she doesn't have the resolution yet to know for sure, but she said these colony-like structures in Alzheimer's disease are often seen when you use silver stain. Now, silver stain is very clumpy and doesn't give you the greatest resolution, but they do stain spirochetes. And when you get a big clump like this, well, maybe that's a whole lot of spirochetes. Other people have seen these plaques. And is this an amyloid plaque that's staining weird in Alzheimer's patients? Or is this Borrelia? Is it syphilis? Well, when we discuss biofilms, here's a biofilm that was grown in culture. But this biofilm came from an Alzheimer's patient, and this is our early work. We still didn't have color stain yet. But this is amyloid plaque, and attached to it are all these Borrelia, and all these little white flecks in here are staining for Borrelia as well. And we call that a Borrelia biofilm, and that is right on top of or mixed into the amyloid plaque. And amyloid plaque is one of the markers we use to determine Alzheimer's disease. If you start doing brain pathology and you look in the hippocampus area of the brain and you find amyloid plaque, you diagnose it as Alzheimer's. Well, that's what they found. But now we have better tools, and this is our stain for Borrelia miyamotoi, and this whole plaque here is amyloid. And you can see the resolution is so much better. We get these nice little spirals that are coming out. And this red that's back here, that's part of the glycofilm, the glycoprotein film that causes the slime layer of the bacteria. So we're not really staining the amyloid plaque, we're still staining the bacteria, and that's why that amyloid plaque looks red. I'll show you another example here. This is Congo red on the left, and that does stain for spirochetes, but it's specifically for the amyloid. And here in this Alzheimer's patient, we picked up the amyloid. But you're seeing that we get these little highlights here that a person can make an argument that those are spirochetes. But when we use the species-specific Borrelia miyamotoi fluorescent stain, we look here, and they stand out a little bit brighter, and then all that red back there, that indicates it's Borrelia miyamotoi as well. Another example, Congo red does pick up the plaque, but here we have these little white inclusions, and then we go over here, the miyamotoi stain, the fluorescent stain, picks up what appears to be spirochetes in better detail. And this goes on and on. Here we have the same white plaque I think we looked at before, but we have spirochetes intermixed into the amyloid. Wherever we find the amyloid and we find Borrelia, the Borrelia is in the form of a biofilm. A biofilm, see, is where the bacteria have a slime layer around them. And when they coalesce as a group, that slime layer kind of mixes together. And then when they get to a big enough group, the center of the slime layer, the biofilm, is protected. It's protected from the immune system. It's protected from the antibiotics we try to destroy it with. It's a place for it to survive better. Now, there's been an argument, let's go back, that these amyloid plaques are actually hunting down pathogens to kill them. And that's been shown in vitro that if you add amyloid and bacteria together in the test tube, the amyloid plaque can kill a variety of bacteria. But it does not kill the Borrelia, and the Borrelia survive, and we can culture it. But we have seen so many cases now of Borrelia miyamotoi in Alzheimer's patients. It's more prevalent, we think, than Borrelia burgdorferi that causes Lyme disease. So maybe one of the main contributing factors of Alzheimer's is actually a relapsing fever. Now, does anyone want to venture why that's bad, besides, dying from it? It's bad because the Lyme disease tests do not detect Borrelia miyamotoi, and we don't really have good tests out there that are commercially available. So when a patient is presenting with Lyme-like symptoms, and they will with Borrelia miyamotoi, you can test over and over and over again with Lyme serology and be negative all the time. Now, has anybody here heard of a patient being tested and retested and they're always negative? Well, somebody pockets that money, but the patient's not getting any benefit from it because they're looking for the wrong thing. Now, bear in mind, we only have 3 stains that stain for 3 species, and we found all 3 species in Minnesota patients. So this Borrelia miyamotoi, which is actually found in spinal fluid, because when this patient, 70-year-old Cape Cod patient, died, they did not send the brain in, but they did have frozen spinal fluid. And we tested that, and look how well it fluoresces. This is a biofilm of Borrelia miyamotoi in the blood. Now this is hopeful because now we have a test that possibly could be useful as a blood test to diagnose some of these dementia patients without waiting for an autopsy. I think most of you would favor that. Here, I have so many slides and I'm going to skip through some of them fast, and this is one of them. I just want to show you we have cytoplasmic DNA markers, and you see how it fluoresces in this way. And this looks over here for the, for the flagellin protein B. We have fluorescent stains that stains Borrelia flagella protein A and flagella protein B. And they always give us this very dense picture of the spirochete. So a very useful fish stain, but it doesn't give us the exact species. But here we have the exact species. This is Borrelia miyamotoi in an Alzheimer's patient. I believe this was a blood smear. And look at the beautiful spirochete out of his blood, and we know the species and we know what it looks like. And so that's what I mean. This is a television picture of the bacteria, whereas a PCR test that just detects DNA and you look at a paper printout in black and white, that's radio. This is color TV. So we're a step above the old technology, and yet it still is not really accepted by the medical community, at least when Lyme disease is concerned. This is a poster that points out some of our pathology, and over here it's available. We accept all donations in form of cash and gold. So now this is not really an Alzheimer's patient, but this is the most important case I'm going to discuss tonight. This is, I became good friends with Scotty over the last 15 years. He was a logger, lived in Twig, Minnesota. He was sick for 30 years. He lives on the edge of a national forest that goes all the way to Canada. He started becoming sick in his mid-30s. He was diagnosed in his late 50s. He tested positive for Lyme disease, Borrelia miyamotoi, and Borrelia mayonii. Scotty was sick for 25 plus years, I guess, and aggressively treated with antibiotics the last decade of his life, and for 7 years, almost continuously with antibiotics, aggressive antibiotic combinations. Okay, and then Scotty did die and had a brain autopsy. His whole family was very supportive. Now, Scotty died of lung cancer. He was a 4-pack-a-day smoker because that's all he could do. But his brother has been sick longer and sick in many worse ways, and was, Chris Shelton was on the list for a heart transplant, and after, well, he had to fight so hard, but after Dr. Papendick treated him for almost a year, his heart shrunk up. He was no longer on that list, and he's doing a lot better. But as you can imagine, you know, he's not 100%. who out here feels 100% after Lyme disease? Well, we're going to talk about maybe why that is. This is where Scotty lived, right here. If you go straight north, you go all the way to the Canadian border. It's all woods. And this 50-acre area here is all open. Everything else, they own like 500 acres, is wooded area. They have a hunting shack right out here by the river, and they have another hunting shack way up here in the National Forest. They've owned dozens of big dogs over the years. Chris's wife has 48 lesions in her brain, and her response to doxycycline almost kills her, so she has chosen not to continue therapy. Chris and Scotty had 3 other brothers. They're all sick. And it's just a tragic story in every way you look at it. And the doctors that have treated them did it entirely on a clinical diagnosis. They did not have the positive test to confirm what they were doing, but they have it now. This is Borrelia burgdorferi found in Scotty's brain. This is Borrelia miyamotoi on the left and Borrelia burgdorferi on the right. And we now call him our control case because virtually every tissue, every sample, every slide had some form of Borrelia in it. And at the end, we're going to talk about the third Borrelia, Borrelia mayonii. So here again, the red is Borrelia miyamotoi, the green is Borrelia burgdorferi. This is the cerebral cortex of his brain. An interesting note is we have never found the spiral form of the bacteria in the white matter of the brain, which tends to be more in the center of the brain by the ventricles. We always find the spirochetal form in the gray matter of the brain, which is the outer reaches of the brain, and the cerebral cortex being one of the main areas, and the hippocampal area another. When you find it in other areas, you usually get a general change of symptoms that a patient has, and we're going to discuss that specifically with the difference between diffuse cortical Lewy body dementia, which is what killed Robin Williams. I mean, yeah, it was suicide, but he was in very rough shape with what he had, and the difference between Parkinsonian presentation of Lewy bodies. And we've worked on both cases, and we have a person in this audience that's very intimately involved with Lewy body dementia. Now I want to put a human face on this, and this is mostly, if there's anyone from the health department, this is what we care about right there. Scotty fought for the last 7 years of his life to make sure people knew how dangerous Lyme was. He would go door to door, talk to his neighbors, put out flyers, had speakers come in, speakers besides me, come in and do talks, and tried to, make the health department stand up and take notice, and I don't know why. This is what it's all about, and he was possibly one of the most gentle, nicest person you could ever meet, and he did not deserve the last 30 years that he got from Lyme disease. Now the third discovery that we're going to talk about is Lewy body dementia, and I'll show you what a Lewy body is in a second here. I'm not going to spend a lot of time on what the symptoms are, but in cortical Lewy body, one of the things that isn't mentioned here is a lot of these patients will rock back and forth, and that just seems to be one of the signatures. But let's see, changes in thinking and reasoning, confusion, alertness varies from one day to another. Parkinson's symptoms might be there, your hunched posture, balance, difficulty walking, shaking, rigid muscles. Visual hallucinations are very common in both forms of Lewy body. Well, actually, cortical Lewy body. Delusions, and trouble interpreting visual information. Like one day they might know what an apple is, the next day they might not be able to tell you what it is. It looks foreign, they can't figure it out. What part of the brain is not picking that up? Trouble, let's see, acting out dreams, basically almost like sleepwalking, and memory loss that can be very significant but less prominent than what you see in Alzheimer's dementia. We're still learning so much about this disease. There's nothing written in stone, but these are just some of the earmarks that we are looking for. So we found in Lewy body dementia, and we've only had a few cases, that Borrelia burgdorferi was associated with the Lewy bodies, and I don't know that we stained for any other bacteria. The Lewy body patients also had nematode parasites in their brain, and we're going to talk about that later. And nematodes is interesting. They share a mimic protein to the neurons. The neurons have a protein called alpha-synuclein, and these Lewy bodies are loaded with alpha-synuclein, but it's slightly different. And we think that affects the human neuron in an adverse way. This is Alan's theory, but it's very interesting how it holds up when you look at it. We think that the Lewy body enters the human neuron, or maybe the human neuron produces the Lewy body. We don't know exactly which, but here's what we're dealing with. Here's a normal neuron up here, and here we see the dendrite going out, and here we see a Lewy body inside a neuron. And eventually, that toxic synuclein protein might kill this neuron. And we think at that point, the neuron wraps itself around this protein, because that protein is, what's the word, dormant, inactive, it's not living, it's just a waxy material that's there. And so when the neuron wraps itself around it, it looks like this, what we call an extracellular Lewy body. It's not inside the neuron anymore. The neuron is wrapped itself around the Lewy body, and all this granular matter here and these spaces, these extracellular neurons always look different than intracellular neurons, and when you stain for specific neuron proteins, they stain. So we think this is what it is. This is a dead neuron wrapped around what used to be an intraneural Lewy body. Now all that sounds, I don't know if you're following that. How many people have I lost? Anyone? I think it's you know, it's going to be posted on a website hopefully, and then you can slow it down and get what you need from it. So, how many feel more confident now than before about beating this disease? This is as scary as you get. Brain autopsy material, autopsies in general, is a scary topic. And I don't mean to make light of it, but I think we've made real headways, and in the end, we're going to show you how some people have maybe benefited from what we've already learned. Here we have, this is a patient from Iowa, and her widow is here today. And these Lewy bodies are pervasive in this patient's brain. But we didn't stain for Lewy bodies. We stained with a red stain for Borrelia burgdorferi. And what's showing up red? The Lewy bodies. So it's probably telling us that Borrelia is intimately associated with these Lewy bodies in some of these patients. That's our conclusion. We need more research, but we need a larger scale study. We can't base broad ranging conclusions on 3 or 4 cases. In Parkinson's disease, if the Lewy bodies are found in a certain area in the center of the brain near the brain stem, you get Parkinson's-like symptoms and you get a different type of Lewy body. The diffuse cortical Lewy bodies are found throughout the outer regions of the brain, the cerebral cortex, the occipital lobe, it can be anywhere, and that can cause a wide variation of symptoms. But when it predominantly is in the hippocampal area, you get a Alzheimer's type presentation. Now the difference is when we see Parkinson's disease, or the center location of Lewy bodies, the protein is not alpha-synuclein, it's a different protein called ubiquitin. And so when we stained with ubiquitin, we found Parkinson's type Lewy bodies in some patients, but mostly we're dealing with the cortical type. Here we have the same Iowa patient I was talking about. Do you mind if I say his name there, Betty? This is Betty Gordon in the front row, and God bless her. She helped fund a lot of this research, not just Lewy body, but almost everything from 2016 that we have done has been because of her donation. When we had these Lewy bodies in Gordon's brain stained, here we used the green fish stain, and you can see that it's lighting up like a Christmas tree. If you just look at that same slide without that stain, you would see virtually nothing. So we're confident that what we're finding, and of course we do run controls, and those controls vary from type and patient. This is an individual spirochete, and this is an extraneural Lewy body down here. But this is probably the most exciting thing, is we found nematodes in the brain of Lewy body patients. And nematodes actually carry a version of the alpha-synuclein. And so we can stain for the alpha-synuclein, and we get these various pictures. This is a stain for pan-cytokeratin, so it picks up the nematodes. But nematodes are a parasite. There's over 2,000 different species. It's the same type of parasite that causes heartworm in dogs. And the fact that it's in the brain gives us hope that there's a two-step treatment. If there's nematodes in the brain, maybe we can treat with an anti-parasite drug. But because the nematodes have in their gut a variety of bacteria, including Borrelia, you probably then have to treat with antibiotics as a follow-up. That's all speculation. But the nematodes are also found in the same tick that delivers Lyme disease. Only a couple researchers have found the nematodes in the tick, but maybe you recognize this name. The first to find it was Willy Bergdorfer. So it's got a historical background. Parkinson's disease with Lewy bodies happens right here. And of course, your set of symptoms when you have pathology in that area of the brain can be very different, very dramatic, and life-threatening. So that's a worse form of this disease probably than the cortical version. Here, the cortical version, that just simply means outer layers. And here we have the cerebral cortex, the temporal lobe, the occipital lobe, and these are all areas that can be affected by Lewy bodies, and each area will give you a different set of symptoms. So that's a very protean disease. You can't really look at a patient's symptoms and say you know what he has, and that's the case with Robin Williams. They kept missing his disease until he died, and then of course it was, from what I understand, he had a very, very advanced case. Here the Lewy bodies stained. We're just staining for Lewy bodies, but here we also stain with special stains looking for Borrelia. Now we go on to the fourth discovery. We're almost over halfway there. Nematode worms were found in 10 consecutive multiple sclerosis spinal fluids. Now, these were obtained from the Multiple Sclerosis Brain Bank in Boulder, Colorado, which is actually owned by the Multiple Sclerosis Society. When we stain specifically for cytokeratin or for alpha-synuclein, we found the worms in the spinal fluid. Now, why hasn't anybody else found this? Well, the first thing is you have to stain with a stain that you would never think of. Cytokeratin is a protein that's never found in the brain, should never be there. So to use that stain is very unusual for a researcher to do. And then, of course, you have to have a positive sample. But the main thing is nematodes are 200 times bigger than Borrelia. So whereas we're using up to 2,000 power to see Borrelia, you're only looking at 40 power to see a nematode. And if you go up above 40 power, it's like taking a magnifying glass and looking at the leg of an elephant and trying to figure out what the whole elephant looks like. You can't do it. So it requires two steps: low power, the right stain, and also the right wavelength of light. And I don't have the patience for that. I am not a good fluorescent microscopist, but I can see these guys with a bright field microscope, and in fact we're doing that work. We are asking for spinal fluid donations. They have to go to a certain facility. Once they're on a fixed slide, then they're sent to me, and then I do the staining. So 10 consecutive multiple sclerosis fluids from Boulder, Colorado, positive for nematodes, consecutive. Well, here we have our publication on the F1000 site, and you can see these are pretty nice pictures. You know, when you get something 200 times bigger than bacteria, and you got all the time in the world to stain them, you get some pretty good pictures. Here's a nematode worm. It's hard to, you know, appreciate this because it's in the brain, but that is really, to me, absolutely gorgeous. And these are stained with ethidium bromide, which is a very toxic stain, and you have to dispose of it as a biohazard. Here we go. Here, this is 2 adult nematodes in the process of mating, and when you mate, eventually you get larval forms of the bacteria. Here's a zygote. I guess I'm going backwards. And here's an egg that's just dividing. And yes, we find eggs in the spinal fluid. This is all spinal fluid work. But we're going to go back and show what we find in the human brain, and besides Lewy body. So now there is a history of finding spirochetes in multiple sclerosis patients' brain. And here again, we only find it in the gray matter, the spirochetes. It goes back to 1911. The main researcher was Dr. Gabriel Steiner, a Jewish German scientist who had to flee Germany in 1933. He probably had the best work ever done on spirochetes and multiple sclerosis. And these are all case studies that were done prior to World War II. And then Gabriel Steiner came to America, but he didn't publish again until 1952. And in 1952, he published a position paper. It's excellent. It just, if you read it, it reads as fresh and wonderful today, and his observations are so clear and accurate. And he made observations of things he didn't even know. Like he said, when spirochetes must be dying, they must shed these round bodies that I call granules. But one of my colleagues thinks those granules are live spirochetes that can convert back to the spiral form. And he put that in his paper back in 1952 and 54. And then, of course, 1957, Rose Eichelson from Philadelphia confirmed his work finding spirochetes in the spinal fluids of MS patients. Then a terrible thing happened. The MS Society came along and in Time magazine said all of this work was nonsense, because if this was an infection, why wasn't it being passed from husband to wife and from sibling to sibling? There's no explanation for that, therefore we think it's a virus. Now we got to think about that. They said in Time magazine they think it's a virus because the spirochete is not passed from person to person. How are viruses spread? So that made no sense at all, but that's how it ended. The 1957 was the end of spirochete research and MS until recently. Dr. Gabriel Steiner, this is what he found in an MS patient in Germany in 1922. And then when he came to America, this is what he found in an Ann Arbor, Michigan patient with MS, and it's a spirochete in the gray matter of the brain. Lyda Matman was his student, and here in 1982 she found a spirochete in the spinal fluid of an MS patient. Now if we're seeing a nematode here, imagine this spirochete here was about 2 inches long in the slide. If that was 200 times bigger, it would be 400 inches long, and that would be what a nematode looks like under the spinal in the microscope. This is Gabriel Steiner's work in 1954, and you can see just a boatload of gray matter spirochetes that he is finding in the brains of MS patients from Ann Arbor, Michigan. Here he's got a close-up of the spirochete half in and half out of neurons. Now, where did we see that before? We saw it in the Alzheimer's patient from Ashland. So I won't dwell too much on this, but you get the point. There's been an association with spirochetes and MS going back to 1911, and we also now see in MS an association with Borrelia, with nematodes. And the nematodes carry spirochetes in their gut. So, is the nematodes delivering this to the brain? We don't know. This is where our research has led, and we want to do more. We want to answer those questions. So you have a speaker coming from Stony Brook, Patricia Coyle, wonderful. She did a study that actually mimics a study we did a year before her. I did a study in Pine County of 26 MS patients, and we treated them with antibiotics for 3 months and then followed them up and looked at seroconversions. And 8 of the patients responded to antibiotics out of 26. That's 15% or so. So in San Francisco in 1996, Pat Coyle presents evidence of 47 MS patients from the New York area, East Coast, and lo and behold, 15% of them had laboratory confirmed confirmation of Borrelia burgdorferi in their spinal fluids, and 13 out of those 15 patients responded to treatment. Now, ironically, she did not, she did not treat the other patients that tested negative. Now, she used a very special test. It's called the antigen capture test of spinal fluid, and it looks, when an antibody connects with a bacterial protein, it forms a complex. And that complex will no longer be registered on a regular blood test. So what she did is she took the spinal fluid, added an alkalizing agent that broke those chemical bonds, and separated the antibody from the protein, and then tested for both, and found both the bacterial protein and the antibody. So that's good research. So we've got the same, almost identical data of 15% of MS patients in a Lyme endemic area seem to carry Borrelia burgdorferi. Okay, the 5th discovery by Alan McDonald is nematode worms were found in Alzheimer patients' brains. And this slide that you're looking at, we stained with pan-cytokeratin with a red dye, and we see a nematode. But we also see some other interesting pathology going on that I'm going to explain in the next slide. But see, this area here is all white. The brain is the most cellular dense organ in the human body. There should be cells everywhere. This whole background should be brain cells, astrocytes, glial cells, neurons, all the brain things that make up brain cells. And we see these white spaces throughout these areas where we find nematode worms. Okay, now these are nematode eggs. So the nematodes the adults are in the brain. These white spaces are the brain cells that they have eaten, and these are the eggs that they're laying in the wake. So now you got a parasite in an Alzheimer's brain laying eggs. And in this, you can see these little purple things. What do you think those little purple bodies are? Well, if a nematode eats, it does indeed, and this is excrement. So we got these huge swaths of cell debris that has been, brain cells have been eaten, it leaves spaces in the brain. We got the eggs, we've got the poop, and we've got nematodes. And here again, if we can catch patients earlier and we treat them correctly, maybe this is a good thing. Maybe we found a new arm of treatment that might be more effective than just guessing with antibiotics. And in this case, we have a non-invasive way of testing, which is the spinal fluid. And that's why we're trying to do the spinal fluid studies now. We want to find out, is this a good way to get earlier diagnosis, a treatment that works, and what it is that we have to do to beat this disease. So here again, this is Alzheimer's brain. There's 2 nematodes in the brain tissue. This is our F1000 paper that I urge you to go to the website and read. And here we have the 6th big discovery by Alan McDonald. Glioblastoma multiforme brain tumor is a fatal brain tumor made up of glial cells from the brain. And we know from the work from the CDC that the Borrelia bacteria has an attraction for glial cells. Now, he was very lucky to get 5 cases because it's a fairly rare brain cancer. And you can find actually several celebrities that have died from this. But he used the fish stains, which were available to nobody else. We're the only ones that had it. And one of the doctors that had this, all 5 samples, he said, these are 5 different patients. I'm interested in, your fish stains. So Alan did the work. And here's the green stain for Borrelia, and these tumors lit up like a Christmas tree. Here again, I wish I had the comparative slide where they did not use the stain, and you can barely see the tumor. You have to stain for tumor proteins, not for Borrelia proteins. But in this case, you see that diffuse throughout the tumor is the evidence of Borrelia burgdorferi, and not Borrelia miyamotoi. This is Borrelia burgdorferi, and I believe that he did stain for Borrelia miyamotoi and did not find that to light up. So we're pretty confident that our stains are species specific and are giving good data. So here we have cases of malignant fatal brain tumors. What's interesting now, we already talked about parasites in the brain, and there was a drug that was recently pulled off the market called mebendazole, very benign anti-parasite drug, still available in the UK for like $2. It is now being repurposed for the treatment of glioblastoma multiforme, and can you guess what the new price is? $230 a day. So mebendazole, by the way, you can find other sources of it at 31 cents a gram. So I would recommend, checking out other sources. So here we go, the 5th case of glioblastoma multiforme, and look at just how bright some of these regions are. Now, when you're looking at a whole portion of the tumor like this, we're only looking at like 100 power. So we're not magnified enough to see the individual spirochetes. Here we have a little more magnification, but you still can't quite see the detail of the individual spirochetes. Then we looked at, this is sort of out of order. These are nematodes in the human brain, but we stained for Borrelia bacteria, and we see that inside the nematode is in fact Borrelia burgdorferi that lights up. And remember what I said before, when you look at other bacteria with fish stains, they appear as little fluorescent dots. And that's what we have here because we're looking at 40 power, not 2,000 power. So here's a much better picture, DNA of the Borrelia bacteria glowing inside a nematode. And this is a multiple sclerosis spinal fluid again. Now, that brings us to our last discovery. And we found 2 species of Borrelia in the human testicle despite 7 years of antibiotics, continuous antibiotics. This is Scotty Shelton's testicle at low power, and we see all the little glowing dots. But here you can actually see the spirochetes. And then we tested for Borrelia mayonii, and here we find it in the testicle. But that's a pretty small picture. And then we have this, that's Borrelia mayonii in the testicle. And these I showed to the Mayo Clinic, and this is what they said was actually not their spirochete at all. It was the edges of human cells that we were detecting. So I offered them our tissue and stain, and just absolutely no return comment, none. So I don't know what to tell you, but we made the offer. We have all that tissue in storage. I mean, it's there, it's not a secret. We want other people to do this work. We're the only ones out there, and if we're the only ones out there, you have to take our word for it. We don't want that. We want you to rediscover what we've already discovered. I was hoping UMD Med School would do that, but not quite. Here we are, the Borrelia mayonii in culture, and this is from Bobby Pritt's work, and I don't know what they used for stain here, but it seems to me a lot of our images look pretty close to what that is. Other pathologies of Lyme disease, we know it can enter, this is a blood vessel, this is Judith McClossy's work, and if it enters the endothelial cells that line this blood vessel, it probably irritates the blood vessel. And if it irritates the blood vessel, the immune system sends out a whole bunch of white blood cells called lymphocytes. And in this case, they were so pervasive, they occluded the entire blood vessel. And this led to the patient's death, who had a stroke because of stoppage of blood flow in the brain. What else kills Lyme patients? This is the 31-year-old patient from New York that had Borrelia in the heart. And I was told this was the Minnesota patient, but I don't think it is. I think this is the same New York patient. But anyway, recently a Minnesota patient also died of cardiac complications of Borrelia, and they found the bacteria in the heart. And they used both types of stains that are, the silver stain is over 100 years old technology, but immunostain goes back about 40, 50 years. The new technology would be our fish stains. You know, back in the day, I was very cautious. I told patients, we can't keep talking about biofilms and cyst forms if we haven't found them. And for, decades, we're talking about, cysts and biofilms and not finding them. Well, this is a biofilm found in a patient. I believe they drained a lymphocytoma from the earlobe, put it on a slide, and lo and behold, this is a biofilm directly from a human patient. So that's from 2007. But prior to 2007, we didn't have a lot of good evidence. We were speculating a lot. And in science, I don't like speculation. I also don't like speaking in black and white terms. And if you went to the early Lyme conferences in 1991, 1992, the people that were the experts at the time were talking in absolute black and white things. Only Borrelia burgdorferi, no other Borrelia. Only the Ixodes damani tick transmit. It's only found in the northeast United States. And it's not transplacental, it's not intracellular, it doesn't get into the brain. The blood tests pick it up. All these things have proven to be untrue. They were never true. And they have not really gone out of their way to re-educate the people they have to. The people that are still operating on 30-year-old information and newspapers reporting that type of thing. But the biggest blunder is assuming that Lyme disease is one disease. It's part of a family of diseases of Borrelia. Relapsing fever, miyamotoi, has proved that the two bacteria can be side by side in the human brain causing problems, and you may never detect it. It's in the brain behind the blood-brain barrier, it's inside human brain cells, and you're dealing with multiple species. Now, a question I'd like to ask the Minnesota Department of Health is if they could name the 4 species of pathogenic Borrelia that are harming patients in Minnesota. We have Borrelia mayonii now, Borrelia basseti, Borrelia burgdorferi, and Borrelia miyamotoi. But what are we testing for over and over again? Just one of those things. We don't have the test for Borrelia mayonii yet. We haven't tried a lot of blood samples yet to know if we can detect it with our fish stains. This is our most recent case. This is a 29-year-old woman in Madison, Wisconsin, a very tragic case. She was bit by ticks in the Ozark Mountains 4 years ago and had a very strange presentation. Didn't get really, really sick until the last year of her life. And we found that she had mostly white matter disease, which is very strange, a white matter form of Alzheimer's specifically. We found amyloid plaque in the white matter. We found also a lot of what's called Lephora bodies, which is very rare. And we found nematodes in the white matter. And then we found Borrelia miyamotoi associated with biofilms and amyloid plaques that were in the gray matter. Here we have the amyloid in her white matter. Oh, and this is amyloid, no, I'm sorry, this is Borrelia biofilm that was from her heart. And this is work done by Paula Pierce at our other lab. And she tested she probably did 50 slides like this. And this is PAS stain. And when you have PAS positive bodies like this, it's probably a Lephora body. And this is, if this stain is very dense that she used, if the stain is lightened up, this is a control slide. This is where you see the Lephora bodies. So now we're seeing Borrelia possibly associated with white matter disease with a whole new presentation and nematode worms involved as well. Now, how are the nematode worms getting around in the brain? And Alan McDonald has a theory on that, that it's entering the spinal fluid somehow, and that it's circulating through the spinal fluid, and then it's most likely to re-enter the brain in the white matter through the ventricles. We don't know. We've designed all kinds of ways to study that. We just don't have the patience, and we don't have the money, and we don't have a large-scale population to do those tests. But this is something certainly on a national level should be done. You know, we should see some CDC dollars dedicated to these kinds of studies. So please help support our research with donations, and we have a donation box back there. And this is an estimated cost of what we spent in the last 2 years. And here's what I want to emphasize here. More than 60% of our donations have come from our own board members. Every time we've dipped into the red, somebody has stepped up to the plate. And Betty here has really gone above and beyond protecting our work. We would have been dead in the water without her. And that's how critical we are of being alive 1 minute and dead the next. Alan McDonald took on $40,000 of personal debt. Other people have contributed thousands and thousands, but the work is very expensive, and all the members of the Paul DeRay Foundation are unpaid. Nobody has ever been paid a paycheck. Even a lot of things like personal expenses, gases, conference, a lot of those aren't. I am going to put in for the conference October 15th because I couldn't go if I didn't. But that's how sparse we are. You get a lot for your money when you donate to the Paul H. DeRay Foundation. And with that, I would like to thank you very much.
My husband has late-stage Lyme disease after a Lyme diagnosis. He has 4 tests, the Identics lab, the eye spot, the advanced lab that says it was finally Lyme disease. He's now at home for memory care, but we have applied to get a brain autopsy done. It's actually through Health Partners in the UM. But we are pretty sure they're not going to be checking for Lyme disease. They will tell us something else. How do we get to you, or how do we get it tested?
You can go to my email, donatebrain@gmail.com. You can request the consent forms, which you just rewrote, because I don't know what they were thinking. Somebody from Madison sent us a frozen brain on dry ice that was completely useless. It didn't have any contact information and didn't have any instructions. And we finally found who sent it to us, and it was a funeral director. And so we've rewritten those instructions to be very clear what we can and cannot accept. But anyway, there's a detail in there for the pathologist or the harvesting physician to contact us. We'll tell them exactly what to do, but it's actually pretty simple. The best and simplest thing to do to solve all the problems is to extract the whole brain, put it in a 5-gallon bucket with brain formalin, let it soak for 3 weeks to firm up, and then ship it Federal Express to our facility in Oklahoma. And then from there, we can prep the tissues in the way that need to be done. That's the simplest thing. Then the next thing would be, what we really like to see is where the pathologist on hand would actually do the biopsies of the brain and put them in paraffin for us. That saves us a lot of time and money. That would be great. We do not, up to this point, by the way, I should mention this, no patient has ever paid for any of the work we've ever done. They did not pay for anything that we did. They did not pay for shipping. We took on all of that. The only thing that we would not pay for is the tissue harvesting, because we have no way of controlling that cost. We have seen people do it for free. Essentia Health did it for free. And we have seen as high as $3,000, which is very unacceptable to me. I would say the average is about $600-700 for a brain autopsy.
$1,000 to do this, and how do we get them to do that and send you the tissue?
Well, first, you want to.
Your email is donatebrain@gmail.com.
That's it.
So we're going to email
And I'll send you the information more detailed, but the best thing is if whoever's holding that tissue for your health partners, if they would have some that's separated and preserved, we can accept formalin fixed, and we can accept paraffin blocks. We no longer accept frozen tissue. And if they would share any of that, that would be great. We'll pay for shipping. But otherwise, contact me and we'll go through the consent forms and get you all set up. But the other thing would be at the time of harvest, if they would send some to health partners and some directly to us. That would be best. So, yes.
I have a question about control groups.
Oh, I'm sorry, I forgot to mention. We used to do free brain autopsies. We ran out of money, so we do have to charge our minimal charge, which is what we pay Paula per hour to prep the slides. Once they're prepped, there is no charge after that. But that averages about $1,200 per brain. So, okay. Sorry that we had to raise the price, but we can't do it if we don't pay the worker. So.
I was going to be speaking a little bit about control groups, because one thing that I've heard from like Dr. Blinker is that most people do have Lyme disease in them, and that not all people express it in Lyme disease. So can you speak a little bit about control groups and like how you do the control groups?
Well, when I talk about controls, we're specifically talking about the sensitivity and accuracy of our stain that we use. Yeah, so I don't know anything about patient populations. We're not in the patient treatment business, but we get asked all the time. You know? Yes?
Can you talk a little bit about nematodes there? Aren't they in the environment anyways? Aren't they found like in water and they talk about them getting into the brain?
Well, that's probably like amoeba histolytica or the other amoeba family that comes in through dirty, stagnant water. It's what killed a lot of the Roman baths, the mud baths they used to take, was the amoebas, got into their brain through their ears. And a lot of those baths are still closed today because they still have the amoebas 2,000 years later.
So a nematode, is that in the tick itself?
Willie Bergdorfer and others have confirmed nematodes in ticks, but nematodes are ubiquitous. They're everywhere. Have you ever eaten a carrot?
Yeah.
Yeah, well, the carrots grow in the soil, and anything that grows in the soil could have a nematode in that soil. So you got to wash it and peel it and make sure and be as safe as you can. But you can pick it up. Dr. Breitschweig says too, never kiss a dog because their mouth is loaded with parasites, including nematodes. So you can pick it up in many ways.
But it doesn't always go to the brain.
Oh no. I mean, most parasites that we talk about infect the GI tract. One of them Giardia. But
That's what I thought. I thought Giardia was.
well, it's not a nematode, but it's a parasite.
Yeah.
And the parasite medicines generally work on a very broad range of parasites. So, yes, you can.
I have a question. Borrelia is a parasite, is that correct?
Well,
Or is it not the same way you're talking about?
it's a sort of a specialized organism. It's similar to malaria, and I believe I would call it a parasite, yeah. It's obligated to the red blood cell, and anything that affects the red blood cell usually gives you high fevers, sweats, chills, shakes, weakness. And, where have we heard that before? That's malaria. So Babesia is like the malaria parasite in the northern hemisphere. So a very, back when I was sick, we didn't really understand co-infections. And boy, I had some pretty high fevers. And I often wonder, was it Babesia? Have no way of knowing. Just, it wasn't in the paradigm of thinking at all. So yeah, it's a parasite, I think. Back there, red.
So you had talked about the channel. Have you tried reaching out to John Hopkins or any other research institutions?
Oh, Alan knows everybody. He knows everybody. He was actually offered to co-author a paper with McCloskey, published through Harvard, and inexplicably, he reacted in a negative way and said, no, I would rather write a letter to the editor. And then Judith McCloskey, out of respect for Alan, backed out. And I talked to her, and I think it was just wonderful that she was so supportive of him because I think his reasons were not good, and he should have participated in that study but he chose not to, and I don't know if Harvard ever did publish that study. But I don't think that's what she'll talk about when she's here, but I'll certainly want to touch base with her and, you know, make sure that we're still on good terms, because her work is incredible. And, you know, one thing that she did and that we've never done is she cultured the bacteria from the Alzheimer brain, but then she took that culture and she created a brain cell culture out of rat brain, and she added the Borrelia she cultured, and within 8 weeks had almost every marker for Alzheimer's. It formed precursor protein amyloid, it cleaved into beta sheet amyloid, she got neurofibrillary tangles, she got hyperphosphorylation of protein tau. You know, what more do you need to support that Borrelia is causing pathology in the brain? And yet, we still hear Borrelia has no correlation to Alzheimer's. And I don't know how that can be said with a straight face. So, yeah, over here.
Is there any comparable work being done like in dogs where you share information with veterinary science people?
I don't know of any veterinarians that are going to this length of pathology. They've certainly done their share of work, which I draw on. We had a great veterinarian, Elizabeth Burgess from Madison, that did great work back in 89, 90. But she was sort of ostracized, and we don't hear much from her. But the veterinarian work is very good, but it's not, to my knowledge, doing anything like what we're doing.
Is there an opportunity to collaborate?
Oh yeah, yeah. We've actually reached out to various veterinarians, and it's very possible that we could share fish stains if they're really set up to do the work. Yeah, we've got 5 fish stains ready to go. I got 3 in my fridge right now. So, but they're $36 a test. So we're broke, but to a big institution who's got a grant, that's nothing. That costs nothing to them. But boy, we have to be frugal. We have to absolutely be, we're talking about like 50 microliters of stain per slide. You know, that's a small amount. Yeah.
If any of us wanted to go get another spinal tap, I know it's not a desirable test, but what would be the test that would be ordered? Because often doctors don't know what to order, you know, and there's so many different ones you can order, so what needs to be ordered that you have to use the doctor?
Well, we ran into this problem just recently. One of our board members, her son was diagnosed, well, first of all, he had Borrelia, he had multiple sclerosis. Then they isolated Borrelia hermsii. Then he had nematodes found in his stool. And then we had a spinal fluid drawn. And the Phoenix Hospital absolutely could not grasp the concept of staining for spinal fluid. They said, we just put it in a machine and it tells us if you have MS. And he said, no, you can't do that. I can, on the phone, I can tell any hospital lab how to do the spinal fluid staining right there. They don't have to send it to us. They've already got all the equipment and stain to do spinal fluids of nematodes. You use a bright field microscope. I mean, the procedure is this. You put a drop on the slide, you let it dry. Then you put the slide in a Copeland jar with 90% alcohol or stronger, and you fix the slide for 30 minutes. Then you take it out, let it air dry. Then you add a small drop of a stain like trichrome or ethidium blue, and most labs will have a automated stainer for trichrome. But then, this is the secret, you have to use bright field microscope with a blue filter start at 4 power. Just the 4 power objective lens. Then you move up to 10 power, 40 power, and you look for it. Ethidium bromide and trichrome blue are excellent if you're not going to use fluorescent microscopy. Ethidium blue will actually work great with fluorescent microscopy, which is like driving a helicopter, by the way. I mean that to be like hard, really hard. But it'll also glow with just blue light, and sometimes if you add a secondary source of ultraviolet light, you get wonderful ethidium blue stuff. But that's a biohazard, and you got to dispose of it in a certain way, and you're supposed to notify the EPA and everything. So we don't use it anymore. We use trichrome mostly. So yes, Betty.
So a
following up on what she just said then, would that your procedure be 100% accurate versus what you and I have talked online where only 10 to 20% of spinal cord tests are accurate?
Well, yeah, I don't know in what context. If we're talking about the finding oligoband proteins and amyloid proteins and antibodies, yeah, probably we're looking at, you know, less accuracy. But the nematodes, I mean, if you find them, that's 100%. I mean, that's visual confirmation. And if it's in the spinal fluid, it's got to be in the brain. And then when we looked at those Borrelia closely, they had, I'm sorry, with those nematodes closely, they had Borrelia inside them. So we're definitely looking at a multi-stage disease. Now, do all multiple sclerosis patients have this? All those same samples from Boulder probably came from the same region. So we don't know. And we might have even picked up, you know, Borrelia hermsii, which is common out there. But, you know, we have to have more tests. But anyway, it's a very simple procedure, and if they were going to ship it to us, they would just take a 5 mils is all we need, a half a tube of spinal fluid, refrigerate it, but it has to be sent to a lab that can accept living samples because they worry about things like mad cow disease and other pathogens. So I can't accept that directly to me. Alan can't accept it directly to him, but we can send it to one of 2 places. And if you contact me on that email, then we can figure out what needs to be done. But I would urge that that hospital just do the staining themselves. Anyone who went through MedTech has got to be able to do it.
Tom,
Yeah.
I just need to interject one comment, and that is that people can't just show up at their doctor's office and request a spinal tap.
Yeah.
The doc's not going to do it because it is an invasive procedure. It does carry risks. So if you're going to have a spinal tap because they've recommended it, that would be great to ask for. But I think a doctor to just say, oh, sure, you can have your spinal tap, that would not be good medicine either.
Yeah, absolutely. All the spinal fluid we got came from patients that have already deceased. They go in and they put a needle in the ventricles and pull it out. So yeah, spinal taps are invasive, and but if you ever have a spinal tap done, ask them for an extra 5 mils. Oh no, that's small. That's small. Sometimes they take 50 mils, so 5 is small. Any more questions?
Yeah, any other questions since we've got Tom here?
It's right here.
Is there any relationship between the Borrelia and any other kind of mental illness like schizophrenia or something?
Well, I know a lot of friends of mine with Lyme disease never pay me back money. It is such a imitator. It's like syphilis. It can mimic a lot of diseases. One of the things is, was idiopathic bladder, interstitial cystitis. That's one that is common. Neurological disease, maybe Parkinson's. We certainly see a Lewy body form of Parkinson's, and Borrelia is involved with that. Maybe ALS, but only, you know, Dr. Marks, who had ALS himself and made a full recovery, or pretty close, recovered on antibiotics, and it was Lyme disease. But he opened a clinic that tested and kept track of all the ALS patients looking for a cure, and he found about 15% responded to antibiotics. The other 85%, you know, who knows what they had or what had to be done. Were nematodes involved there? We don't know. The work's never been done. But yes, there's many neurological and peripheral diseases that I think are caused by Borrelia, but you don't know until you discover it, you know, and prove it. You know. But it's boy, this is a pervasive bacteria. It's modal. It gets through the whole body, infects any organ. So who's to say if you have an adrenal problem that, you know, the adrenal gland hasn't been affected? I don't know. That's a lot of money to study a lot of things. You know.
Is it worse than Zika?
Well, here, that's apples and oranges. I'm worried about Zika. I wasn't, you know, a year ago, but I think, you know, it's creeping up. It's in Florida. Yeah, Zika is a big deal. So, you know, and who hasn't been bit by mosquitoes? If you go to Florida, how are you ever going to avoid getting bit by a mosquito? So she knows the answer.
Can you test a live person's brain? I mean, I'm just curious.
Only
Is
if,
there any way to like do a biopsy on a living?
well, there is. That's pretty invasive. I'd say that's the most invasive thing. Most people are not lining up for a brain biopsy. But if you were having, like these glioblastoma patients, if they wanted to take extra biopsies of the brain when they're in there, yeah, we could look at that point. But you know, what I'm presenting to you is not really widely known. It seems, you know, amongst the chosen few, this is old information, but the general medical population don't really know this work. And we don't know how to get through. You know, we have not been accepted in any major medical journals. Alan turned down Harvard and the Journal of Infectious Diseases. So I don't know. That would have been really, I think, our big breakthrough.
Is the white matter the center part and gray is the outside part?
The white matter is the neurons that are coated with myelin, and it's an insulation, and it makes that part of the brain look white. And it's mostly in the central part of the brain surrounding the lateral ventricles. And for some reason, when the spirochetes get there, they might take on a different form. They might be the granular form. Actually, Dr. Gabriel Steiner in 1954 mentioned that they only found the granular form in the white matter. They didn't find the cortical spirochetes or the classical forms. So, I saw some over here. Questions? Betty.
Yeah, he had his hand up. I do want to talk just briefly.
All right.
Go ahead.
Was it- Yes.
Is there a vaccine? They used to have a vaccine, I talked to people who had a vaccine that somehow can.
I hope we never see that vaccine ever again. They're talking about you know, it's interesting, when Lymerex came out, you know, 3 class action lawsuits came out. There's people in this room I could point out that had very adverse reactions to that. And what's interesting, when they tried to relaunch it, one of their press releases said, Lymerex never had any adverse reactions in any patients, but just in case it did, we changed the molecule and got rid of the lipid moieties so that this will have less side effects than the original vaccine that never had any side effects. Now, if you can make sense of that, I don't know. But I never want to see an OSPE vaccine on the market again. We'll never see the European vaccine, decorin binding protein, and OSPC, and some of the others that were quite successful in animals. I would like to see those, but we don't. There is a dog vaccine that's both an OSPE vaccine and then a separate one by Fort Dodge that is a bacterium, which I actually prefer. They took, you know, like 30 species of Borrelia, boiled them down, put them in a shot, and it's good for about 4 months, and the dog is covered. But OSPE in humans has just proven to be a disaster, in my opinion. And going in 2 years before, I signed a petition against it that was put together by Dr. Miller, the psephologist, who had all his graduate students put the petition together. And it was based not on, you know, some of the evidence that's come out now about OSPE, but it was based back then that if you were exposed to Lyme disease, outer surface protein A accumulated in joints. And if you got the vaccine and you made antibodies against OSPE in your joints, we were concerned you would get joint arthritis. And that's what happened in animal models. Dr. Ron Schell in Madison showed that with mice. And so we all signed this position paper saying, you know, don't market this, don't approve it. Well, it got approved. And they rushed it to market because an identical vaccine from Connaught Labs was in the market, and they wanted to beat them out. Well, when the vaccine came out, it proved to, we think, support what we were saying. But now there's more evidence that shows that the lipid portion of the molecule actually affects the immune cells in an adverse way, and that might lead to a chronic infection when you get the Lyme disease. So I don't know. The data is pretty sparse, but my personal opinion is the people that are behind the original OSPE Lymerex vaccine are not people that I would invite to my party. So, and this is Betty Gordon from Iowa, and her husband Jack Gordon had the cortical Lewy body dementia, our first case in entree into that disease. So she wants to say a few words here.
Yes, hi. First of all, I cannot get over your support group. I came last year and you had a full house, and I really applaud you for really getting involved and taking active participation. Briefly, a year ago, I told you at that time I had submitted my husband Jack Gordon's brain to Paula down in Oklahoma, and then it went to Alan McDonald. And as Tom just said, my husband's brain made worldwide history, being the first one with these two diseases being ever found together before, the Borrelia Lyme disease and Lewy body dementia, causing the very violent hallucinations that Robin Williams also had. And Alan McDonald had told me at the time, Betty, he's the first. We're going to work on getting him published in a scientific medical journal somewhere in the future. And so I was looking forward to that. Now get this, I'm a non-medical person, but I'm going to be the first co-author if this ever takes place over Alan McDonald, Paula Pierce, and Tom Grier here. But I got involved on this, well, Tom and I go back 10 to 12 years talking online, but it was last year I met him for the first time. But okay, a year ago or something, they were doing a fundraiser for Alan on his research and telling all about what he was going to do. And I've been so impressed by the free videos that he's done for so many years of making things simple, you know, Lyme friendly. So we didn't have to have medical degrees to understand all the terminology being used. So at the time, I decided I was going to send him $1,000 contribution, because at the time, they were saying it took $1,000 for one person to do the blood staining involved on this. Well, wouldn't you know, then later on, Jack passed, it was quite a sudden thing, because he died of cancer 2 nights after being diagnosed with cancer in the lung and the liver. And, you know, I'm still shocked at this point, going on 2 years later. But anyway, I had this brain autopsy done because I wanted the truth of scientific evidence. What happened? Were the doctors right? And no, they weren't. They never, Jack was misdiagnosed for 35 years, as well as myself. Never diagnosed or treated for the Lyme disease or his Lewy body dementia. And I wanted peace of mind of knowing what he had. And this has given me that, of what you and Alan and Paula have done, that no one can ever take away from. That I know what he had and so on. And because of that, I gave an extra $40,000 for them to do this. And being frugal, that was Jack and I, because we had lower paying jobs and so on, so this was a big step for me to take. But I wanted other families to feel the way I feel of having gotten this information. This is all coming from the heart and everything, so, but it's just great knowing this and so on, that others can be helped the way I have been. And in turn, in Iowa, I'm trying to get Jack's death certificate amended, showing that he had died from the Lyme disease and the Lewy body dementia contributed to his death. Well, boy, is that fighting a big cause again. That's like taking our bill to Congress, folks. Because in turn, I'll need a court order in order to do this. I need to go through either my county or state coroner to do this. And I need an actual death certificate from Alan McDonald, since he had done the work on this, to do it. And we had sent him a poster board that would be available also on the Jury site and so on, describing him and the other Cape Cod Lewy body dementia folks. That wasn't good enough. It's got to be an actual death certificate. I'm also trying to get him added to Iowa's Lyme CDC statistics. Well, you know how that goes too. So it's been really quite the thing, but I encourage you to think about this, and to the wife out here who's going through the same thing too, of how having the power of attorney and so on, that type of thing, and getting involved and knowing for yourself what did he have and not have. Because before Jack got suddenly sick in 2014, he says, Betty, I want to have a serious talk with you. And so we did for about an hour. And with Jack at that time, with the Lewy body dementia being so bad, where he didn't know a whole lot what was going on. I didn't know what was coming. But the first thing out of his mouth was, Betty, I want to go and live by myself. Can you imagine what that feels like when you've been helping him all this time of getting in and out of bed, you know, lifting him up, walking backwards from room to room and all that sort of thing. But I realized too, that was his way of telling me he was ready to go to heaven. You know, he had no quality of life. My husband was a jack of all trades, of anything and everything, helping people galore. But towards right before he died, one night when he was very lucid, he says, well, Betty, I love you more than you'll ever know. And I cannot thank you enough for taking such good care of me. And so that made up for the other incident and so on. But anyway, I thank you for hearing me out, and I encourage you to get involved and do what you can to help on the jury thing too, because they've given, as you know. And I didn't know too that Ellen had been paying out of pocket for all this stuff, but I'm very glad to have contributed what I could and to have made a difference. Thank you.